Hello everyone, I am Nikita and today's topic we are going to discuss about the DNA translation process. What is the DNA translation process? Is the process in which the sequence of codon on the mRNA strand is used and accordingly the amino acid are joined together to form a polypeptide chain that makes a proteins. Then the process involves the following steps. First, activation of amino acid and formation of amino acid tRNA complex and the formation of the polypeptide chain. So first, in the presence of enzyme amino acyl tRNA synthetases, the amino acid molecule is activated and each then amino acid is attached to the specific tRNA molecule at the 3' end to form a amino acyl tRNA complex. This reaction needs ATP. Then formation of polypeptide chain are divided into three steps: initiation, elongation and the termination. So first, initiation. It begins with the formation of initiation complex which requires a mRNA codon for the polypeptide, the smaller 30S and the larger 50S subunit of ribosomes. The initial amino acyl tRNA complex, ATP and GTP are as a source of energy. Then the presence or the process of initiation needs initiation factors. In prokaryotic, the first amino acyl tRNA complex has amino acid N-formyl methionine, while in eukaryotic it's methionine. The process starts with the binding of mRNA on the smaller 30S subunit ribosomes. The start codon AUG its position is properly. The amino acyl tRNA complex, that is F-met tRNA complex, now gets attached to the start codon AUG. This is done with the help of the anticodon. Small and large unit of ribosome join to form a 70S ribosome. The ribosome has three sites, namely amino acyl site, peptide site and exit site. The empty tRNA leaves exit sites only. Then elongation. After initiation process, the elongation process starts. This is done by formation of the peptide linkage bond between the successive amino acid molecules. The elongation activity is catalyzed by the enzyme peptidyl transferase, each tRNA complex being a specific amino acid. Due to complementary nature of anticodon and the codon, the amino acid are placed in their proper positions. The elongation activity involves the elongation factor. During elongation, the ribosome moves along the mRNA in stepwise manner from start to stop codon. One codon ahead each time. This movement is called translocation. In every step of translocation, one amino acid is added in the polypeptide chain using elongation. Then termination. Elongation continues until the ribosome adds the last amino acid coded by the mRNA. When the ribosome reaches to the last termination codon in either UAA, UGA or the UGA, termination occurs. It identifying the termination codon and releasing the polypeptide chain from the side. The release or the termination factors R1, R2 chain from the site release or the terminate factor R1, R2, R3 play an important role. The termination, the smaller 30S and larger 50S unit of ribosome get separated from each other. The energy requirement for the polypeptide or the protein synthesis activity is fulfilled by the ATP and the GTP molecules. Thank you.